Prelude to the subject of DNA nanotechnology, we're going to talk about the self-assembly of the DNA double helix itself. The molecule is illustrated schematically at the top of the slide and in chemical detail down here. So this ribbon-shaped structure that you see spiraling around is one half of the double helix, and it is comprised of a series of sugar molecules comprised of carbon and oxygen that are coupled together via phosphate groups. So looking at this, you see one ribbon here, one ribbon here. The whole thing winds up because of the constraint of packing these groups while having these groups stacked nicely on top of one another to form a helical structure like this. The key observation of Watson and Crick was that these combinations of the bases, thymine with adenine, guanine with cytosine, resulted in exactly the same distance from sugar to sugar, and therefore a somewhat uniform molecule could be self-assembled by the hydrogen bonding scheme shown here. Let's look at this in yet more detail. The sugar rings are where the D comes from for deoxyribose. You see there's an oxygen here in the backbone pendant to the sugar. In ribose nucleic acid, which some of you may have heard of as RNA, there's an additional oxygen on the ring right here, and that affects the mechanical and chemical properties of the polymer quite a lot. The second thing to notice on this backbone is that the oxygen pendant from this phosphate here has only one bond, so this group is negatively charged. So every 3.4 angstroms, there's another negative charge and one across the helix over here. DNA is what is called, at least in water, a polyelectrolyte because it's a long charged polymer, and the A stands for acid, deoxyribose nucleic acid, and the acid comes about because of the negative charges on these phosphate groups. It's like a phosphoric acid. Okay, a third thing to notice about the backbone is it has a directionality. That is, if you look at the plane of these sugar molecules with the oxygen atoms below, then they're all stacked this way going along this chain, and they're stacked the opposite way going along the other side. This gives DNA an inversion symmetry, a C2 symmetry, that it wouldn't have if the chains didn't run in opposite directions, and this was part of the solution of the structure for Watson and Crick when they saw the X-ray patterns generated by experimentalists. Now let's turn to the bases. Actually, the bases can both donate or accept protons, so in the classical sense they're not really bases. The nitrogens in the bases, though, mean that they can donate electrons from the lone pair, and this is the Lewis definition of an acid or a base. So this is why they're called bases. In a sense, donating an electron is the same as accepting a proton. Let's look at these structures. We already talked about adenine and thymine and the formation of hydrogen bonds, and the fact that you have in this oxygen right here an acceptor, and in this NH group right here a donor. In the case of guanine and cytosine, you have an acceptor on the guanine, a donor from this NH group here, and an NH2 group here which also presents two donors actually. And over here an oxygen acceptor, a nitrogen without a hydrogen as an acceptor, and an NH2 group here as a donor. So it's this specific pattern of hydrogen bonded pairing, donor with acceptor in the right order, in the right spatial position, that leads to self-assembly of the double helix if the code on one strand, TCAG, sees its partner, A with T, G with C, T with A, C with G, on the opposite strand. It will then self-assemble. So I guess the final thing that needs to be said about this is that we're going to draw DNA without worrying about the helical structure. So we'll maybe draw a line like this and A, T, C, G. We're going to look at structures in this form, A pairing with T, C pairing with G, just by drawing lines like this. It turns out that the association energy of the hydrogen bonds is on the order of 5 kilocalories or so per mole. We talked about this before. So they break relatively infrequently as single hydrogen bonds. on millisecond timescales, or single hydrogen bonds perhaps on microsecond timescales. But if we now zip a row of them up together, their free energies are additive, and so the dissociation constant becomes greatly decreased. And the result is that the double helix becomes thermally stable if enough bases are joined together. The key number turns to be about, on average, 8 base pairs at 300 Kelvin. For the double helix to remain stable for long times, obviously the more G's and C's you have, the more triply hydrogen bonded motifs, the more stable the double helix. So this is sequence dependent. Oh, I want to say one other thing. The acid part, of course, is highly soluble, the phosphates on the outside. So these need to face the water. The bases themselves have these organic rings that are not particularly soluble in water. In fact, they're hardly soluble at all. And furthermore, they like to stack on top of one another. So there is a stacking interaction between bases, and they exclude water. These interactions drive the bases into the inside of the double helix, where they hydrogen bond, while the phosphate groups interact strongly with water and help to dissolve the double helix, driving the self-assembly of the double helix. One final word. There's a lot of negative charge facing a lot of negative charge here, so these base stacking and hydrophobic interactions that keep the bases in the interior can be pushed apart by Coulomb repulsion between these phosphate groups. This is overcome by having excess salt in solution, which not only balances the charge on the oxygens, but screens the Coulomb interaction, as we learnt when we derived the Debye length in Chapter 3.